So in this video we are going to learn about urinary system. The urinary system consists of two kidneys, two ureters, one urinary bladder and one urethra. So functions of the urinary system: the kidneys regulate blood volume and composition, help regulate blood pressure, synthesize glucose, release erythropoietin, participate in vitamin D synthesis and excrete wastes in the urine. So the ureters transport urine from the kidneys to the urinary bladder. The urinary bladder stores urine. The urethra discharges urine from the body. And along with that, kidney functions. The kidneys do the major work of the urinary system. Functions of the kidneys include the following: regulation of blood ionic composition, regulation of blood pH, regulation of blood volume, regulation of blood pressure, maintenance of blood osmolarity, along with production of some hormones like calcitriol and the the active form of vitamin D and erythropoietin. Then regulation of blood glucose level along with excretion of wastes and foreign substances. Now we are going to learn about the anatomy of the kidney. The paired kidneys are reddish kidney bean-shaped organs located between the peritoneum and the posterior wall of the abdomen. The right kidney is slightly lower than the left because the liver occupies considerable space on the right side superior to the kidney. So, right kidney is slightly lower than the left. Then external anatomy of the kidney. 3 layers of tissue surround each kidney. The deep layer, the renal capsule, is a smooth, transparent sheet of dense, regular connective tissue. Renal capsule is a smooth, transparent sheet of dense, regular connective tissue that is continuous with the outer coat of the ureter. It maintains the shape of the kidney. The middle layer, the adipose capsule, first renal capsule, along with that adipose capsule, is a mass of the fatty tissue surrounding the renal tube capsule. It also protects the kidney and holds it firmly in place within the abdominal cavity. The superficial layer, the renal fascia, is another thin layer of dense, irregular connective tissue that anchors the kidney to the surrounding structure and to the abdominal wall. So, 3 layers are there, like renal capsule, adipose capsule, along with that renal fascia. Now about internal anatomy of the kidneys. A frontal section through the kidney reveals 2 distinct regions: a superficial smooth structured or textured area called the renal cortex and a deep reddish brown inner region called the renal medulla. The renal medulla consists of cone-shaped renal pyramids. The base of each pyramid faces the renal cortex and its apex, called the renal papilla, points towards the renal hilum. So renal medulla, renal cortex regions are there. Along with that, the renal medulla consists of cone-shaped renal pyramids. The base of each pyramid faces the renal cortex and its apex, called the renal papilla, points towards the renal hilum. The renal cortex is divided into an outer cortical zone and an inner juxtamedullary zone. Those portions of the renal cortex that extend between renal pyramids are called renal columns. Then together, the renal cortex and renal pyramids of the renal medulla constitute the parenchyma, functional portion of the kidney. Within the parenchyma are the functional units of kidney called nephrons. The hilum expands into the cavity within the kidney called the renal sinus, which contains part of the renal pelvis, the calyces and branches of the renal blood vessels and nerves. Let's learn about its structure. We could see portions like renal cortex region, along with that renal medulla, then renal columns, renal pyramid in renal medulla, then renal sinus along with renal papilla, then fat in renal sinus, then renal capsules are there, then renal artery, renal vein, then some regions like renal capsule. So nephron, path of urine drainage, collecting duct, primary duct in renal pyramid, then minor calyx, then major calyx, renal pelvis, ureter, urinary bladder. So this shows the frontal section of right kidney. We know that right kidney, situated a bit lower than the left one. Then blood supply of the kidneys. The kidneys receive 20 to 25% of the resting cardiac output via the right and left renal arteries. Within the kidney, the renal artery divides into several segmental arteries which supply different segments or areas of the kidney. Each nephron receives one afferent arteriole which divides into a tangled ball-shaped capillary network called the glomerulus. The glomerular capillaries then reunite to form an efferent arteriole that carries blood out of the glomerulus. Glomerular capillaries are unique among capillaries in the body because they are positioned between two arterioles rather than between an arteriole and a venule. They are positioned between two arterioles. That is a special case. But other blood capillaries are positioned between an arteriole and a venule. But glomerular capillaries are unique. They are positioned between two arterioles, afferent and efferent arterioles. So the efferent arteriole divide to form the peritubular capillaries. Extending from some efferent arterioles are long loop-shaped capillaries called vasa recta. The peritubular capillaries eventually reunite to form peritubular venules and then interlobular veins. So blood leaves the kidney through a single renal vein that exists at the renal hilum and carries venous blood to the inferior vena cava. So the glomerular capillaries then reunite to form an efferent arteriole that carries blood out of the glomerulus. Glomerular capillaries are unique among capillaries in the body because they are positioned between two arterioles rather than between an arteriole and a venule. Then the efferent arteriole divide to form the peritubular capillaries. Then extending from some efferent arterioles are long loop-shaped capillaries called vasa recta. The peritubular capillaries eventually reunite to form peritubular venules and then interlobular veins. Blood leaves the kidney through a single renal vein that exists at the renal hilum and carries venous blood to the inferior vena cava. Then next topic is about nephron. Nephrons are the functional units of kidneys. Each nephron consists of two parts, a renal corpuscle where blood plasma is filtered and a renal tubule into which the filtered fluid passes. The two components of a renal capsule are the glomerulus capillary network and the glomerular capsule or Bowman's capsule. A double-walled epithelial cup that surrounds the glomerular capillaries. Blood plasma is filtered in the glomerular capsule and then the filtered fluid passes into the renal tubule. Then the renal tubule consists of a proximal convoluted tubule, loop of Henle, nephron loop, and distal convoluted tubule. Convoluted means the tubule is tightly coiled rather than straight. Then the distal convoluted tubules of several nephrons empty into a single collecting duct. Collecting ducts then unite and converge into several hundred large papillary ducts which drain into the minor calyces. The collecting ducts and capillary ducts extend from the renal cortex through the renal medulla to the renal pelvis. So in a nephron, the loop of Henle connects the proximal and distal convoluted tubules. The first part of the loop of Henle dips into the renal medulla where it is called descending limb of the loop of Henle. It then makes that hairpin turn and returns to the renal cortex as the ascending limb of the loop of Henle. About 80 to 85% of the nephrons are cortical nephrons. Their renal corpuscles lie in the outer portion of the renal cortex and they have short loops of Henle that lie mainly in the cortex and penetrate only into outer regions of the renal medulla. The other 15 to 20% of the nephrons are juxtamedullary nephrons. Their renal corpuscles lie deep in the cortex close to the medulla and they have a long loop of Henle. So, overview of renal physiology. To produce urine, nephrons and collecting ducts perform three basic processes like glomerular filtration, tubular reabsorption and tubular secretion. Then glomerular filtration. In the first step of urine production, water and most solutes in blood plasma move across the wall of glomerular capillaries into the glomerular capsule and then into the renal tubule. So glomerular filtration is the first process. In the first step of urine production, water and most solutes in blood plasma move across the wall of glomerular capillaries into the glomerular capsule and then into the renal tubule. Then tubular reabsorption. As filtered fluid flows along the renal tubule and through the collecting duct, tubule cells reabsorb about 99% of filtered water and many useful solutes. Then tubular secretion. As fluid flows along the renal tubule and through the collecting duct, the tubule and duct cells secrete other materials such as waste, drugs, and excess ions into the fluid. Then, first step, we are going to elaborately learn that topic, like glomerular filtration. The fluid that enters the capsular space is called the glomerular filtrate. On average, the daily volume of glomerular filtrate in adults is 150 liters in females and 180 liters in males. More than 99% of glomerular filtrate returns to the bloodstream via tubular reabsorption, so only 1 to 2 liters are excreted as urine. Otherwise, it would be very difficult. Don't you think? That's why reabsorption process is very important. Net filtration pressure. Glomerular filtration depends on three main pressures. One pressure promotes filtration and two pressures oppose filtration. So glomerular blood hydrostatic pressure. Glomerular blood hydrostatic pressure, GBHP shortly, is the blood pressure in glomerular capillaries. Generally, GBHP is about 55 mmHg. It promotes filtration by forcing water and solutes in blood plasma through the filtration membrane. Then capsular hydrostatic pressure, CHP, is the hydrostatic pressure excreted against the filtration membrane by fluid already in the capsular space and renal tubule. CHP opposes filtration and represents a back pressure of about 15 mmHg. Then blood colloid osmotic pressure, BCOP, which is due to the presence of proteins such as albumin, globulin, and fibrinogen in blood plasma, also opposes filtration. The average BCOP in glomerular capillaries is 30 mmHg. Net filtration pressure, the total pressure that promotes filtration, is equal to GBHP minus CHP is minus BCOP. NFP is equal to, that means net filtration pressure is equal to, we will be subtracting those values like 55 mmHg minus 15 mmHg minus 30 mmHg. We will get 10 mmHg. So that is the NFP rate, like net filtration pressure. Not rate, net filtration pressure, that particular value. Thus, a pressure of only 10 mmHg causes a normal amount of blood plasma minus plasma proteins to filter from the glomerulus into the capsular space. Then, next topic, glomerular filtration rate. The amount of filtrate formed in all the renal corpuscles of both kidneys per minute is the glomerular filtration rate. The amount of filtrate formed in all the renal corpuscles of both kidneys per minute is the glomerular filtration rate, GFR. In adults, the GFR averages 125 ml per minute in males and 105 ml per minute in females. The mechanism that regulates glomerular filtration rate operates in two main ways: by adjusting blood flow into and out of the glomerular apparatus like glomerulus, and by altering the glomerular capillary surface area available for filtration. GFR increases when blood flow into the glomerular capillaries increases. Three mechanisms control GFR, like renal autoregulation along with neural regulation, then hormonal regulation. Next, tubular reabsorption and tubular secretion. Tubular reabsorption is a selective process that reclaims materials from tubular fluid and returns them to the bloodstream. Reabsorbed substances include water, glucose, amino acids, urea, and ions such as sodium chloride, potassium bicarbonate, and phosphate. Some substances not needed by the body are removed from the blood and discharges into the urine via tubular secretion. This include urea, creatinine, and certain drugs. So this picture depicts this info in a detailed manner, like summary of filtration reabsorption and secretion in the nephron and collecting duct. Filtration occurs in the renal corpuscle. Reabsorption occurs all along the renal tubule and collecting ducts. So first process, renal corpuscles. And we zoom this like this. You could see that portion. Renal capsule. So that renal capsule is visible here. So glomerular filtration rate about 105 to 125 ml per minute of fluid, that is isotonic to blood. Filtered substances, water and all solutes present in blood including ions, glucose, amino acids, creatinine and uric acid. So that is something happening in the renal corpuscle located in this region, which is represented with the alphabet A. Then next, proximal convoluted tubule. Reabsorption of filtered water, Na plus, K plus, glucose, amino acids, Cl minus, HCO3 minus, urea, Ca2 plus, Mg2 plus, etc. Then secretion into urine of H plus, NH4 plus, urea and creatinine secreting. That portion secretes this contents like H plus, NH4 plus, urea, creatinine into the urine. That is something happening in the proximal convoluted tubule, which is represented with the help of alphabet B in this picture. Later, loop of Henle. Reabsorption of filtered water, Na plus, K plus, Cl minus, HCO3 minus, Ca2 plus, Mg2 plus and urea, and along with that secretion of urea in the C region, loop of Henle. Then later D, like distal convoluted tubule, which is represented with the help of alphabet D as shown in this picture, like reabsorption of water, Na plus, Cl minus, Ca2 plus, etc. Then principal cells in late distal tubule and collecting duct. They carry out reabsorption of water, Na plus, urea and secretion into urine of K plus. Then intercalated cells in late distal tubule and collecting duct. Reabsorption of HCO3 minus and urea, secretion into urine of H plus. That is happening in this intercalated cells in the late distal tubule and collecting duct. F represented in the collecting duct region. Then hormonal regulation of tubular reabsorption and tubular secretion. 4 hormones affect the extent of Na plus, Cl minus and water reabsorption as well as K plus secretion by the renal tubules. The most important hormonal regulator of electrolyte reabsorption and secretion are angiotensin 2 and aldosterone. The major hormones that regulate water reabsorption is antidiuretic hormone, then renin, angiotensin, aldosterone system. When blood volume and blood pressure decrease, the juxtaglomerular cells secrete the enzyme renin into the blood. Renin clips off a 10 amino acid peptide called angiotensin 1 from the angiotensinogen, which is synthesized by hepatocytes. By clipping off 2 more amino acids, angiotensin converting enzymes converts angiotensin 1 to angiotensin 2, which is the active form of the hormone. So angiotensin 1 is about 2 more amino acid short than angiotensin 2. No, angiotensin 1 has 2 amino acids greater than this angiotensin 2. So this is formed with the help of clipping of 2 amino acids. Later angiotensin 2 is formed, which is the active form of the hormone. Then angiotensin 2 affects renal physiology in 3 main ways. Like it decreases the glomerular filtration rate by causing vasoconstriction of the afferent arteriole. It enhances reabsorption of Na plus, Cl minus and water in the proximal convoluted tubule. Then it stimulates the adrenal cortex to release aldosterone, a hormone that in turn stimulates the principal cells in the collecting duct to reabsorb more Na plus and Cl minus and secrete more K plus. Then antidiuretic hormone or ADH. Antidiuretic hormone releases released from the posterior pituitary plays an essential role in this process by increasing collecting tubule permeability to water, which is very low in the basal state. ADH appears to act by inserting aquaporin 2 water channels into the luminal membrane, thereby allowing transcellular water reabsorption to occur down an osmotic gradient. Then urine transportation, storage and elimination. From collecting ducts, urine drains through papillary ducts into the minor calyces, which join to become major calyces that unite to form the renal pelvis. From the renal pelvis, urine first drains into ureters and the urinary bladder. Urine is then discharged from the body through the single urethra. Each of the two ureters transports urine from the renal pelvis of one kidney to the urinary bladder. Peristaltic contractions of the muscular walls of the ureters push urine towards the urinary bladder. Then the urinary bladder is located in the pelvic cavity. Its function is to store urine before micturition. The micturition reflex discharges urine from the urinary bladder via parasympathetic impulses. The urethra is a tube leading from the floor of the urinary bladder to the exterior. Its anatomy and histology differ in females and males. In both sexes, the urethra functions to discharge urine from the body. In males, it discharges semen as well. Then, characteristics of normal urine: color yellow or amber color. Color is due to urochrome, like that color is due to urochrome, pigment produced from breakdown of bile, urochrome, and urobilin from breakdown of hemoglobin. Breakdown of bile and breakdown of hemoglobin, urochrome and urobilin. Color mildly aromatic. Odor mildly aromatic, but sometimes ammonia-like upon standing. The urine of diabetics has a fruity odor due to presence of ketone bodies. Then pH ranges between 4.6 and 8.0, average 6.0. Varies considerably with diet. High protein diets increase acidity, vegetarian diet increase alkalinity. Then specific gravity in urine, it ranges from 1.001 to 1.035. The higher the concentration of solutes, the higher the specific gravity. Next, abnormal constituents in urine. The presence of glucose in the urine is called glucosuria and usually indicates diabetes mellitus. The presence of red blood cells in the urine is called hematuria and generally indicate a pathological condition. High levels of ketone bodies in the urine called ketonuria may indicate diabetes mellitus, anorexia, starvation, or simply too little carbohydrate in the diet. Ketone bodies increase, ketonuria increase, which shows anorexia or diabetes mellitus, starvation, or simply too little carbohydrate in the diet. Then formation of concentrated urine. The three major solutes that contribute to its high osmolarity are Na plus, Cl minus, and urea. Two main factors contribute to building and maintaining its osmotic gradient, like differences in solute and water permeability and reabsorption in different sections of the long loop of Henle and the collecting duct, and countercurrent flow in opposite directions of fluid in neighboring descending and ascending limbs of the loop of Henle. Then countercurrent mechanism is responsible for generation of osmotic gradient in the renal medulla. The basis of this mechanism lies on the hairpin shape of the long loop of Henle of juxtaglomerular nephrons. The descending limb of loop of Henle carries tubular fluid from the renal cortex deep into the medulla, and the ascending limb carries it in the opposite direction. Thus, fluid flowing one tube runs counter opposite to fluid flowing in a nearby parallel tube, an arrangement called countercurrent flow. Then countercurrent exchanger, a system in which heat or chemicals passively diffuses across a membrane, separating two countercurrent exchanger streams so that at each end the fluid leaving along one side of the membrane nearly resembles the temperature or composition, the fluid entering the other. The venae commits in the arms serve as a countercurrent exchanger, the arterial blood serving to rewarm the cooler venous blood. Then countercurrent multiplier, a system in which energy is used to transport material across a membrane, separating two countercurrent multiplier tubes connected at one end to form a hairpin shape. By this means, the concentration can be achieved in the fluid in the hairpin bend relative to the inflow and outflow fluids that is much greater than the transport mechanism could produce between the two sides of the membrane at any point. The nephronic loops in the renal medulla act as a countercurrent multiplier. Then osmoregulation is the active regulation of osmotic pressure of bodily fluids to maintain the homeostasis of the body. It regulates the concentration and osmotic pressure of blood by regulating the water currents of blood plasma. It is an important process as excess loss of water may cause dehydration, whereas excess of water intake may dilute the body fluids. The human kidney is extremely flexible in its working. It excretes large amount of hypertonic urine when water intake is very high, while it excretes small amount of hypertonic urine when water is deficient and needs to be conserved. So in this lecture we learned about urine formation mechanism, properties of urine along with the structure of kidney, proximal convoluted tubule, distal convoluted tubule, loop of Henle, glomerulus, and how urine is formed. Three processes like filtration, glomerular filtration, tubular reabsorption, and then secretion. And along with that, countercurrent multiplier system, osmoregulation process, then countercurrent exchange system. So that's all about this topic. Thank you for listening. We just had a quick recap of all this kidney related topics of urinary system. So thank you for listening.